very exciting and an interesting session. Who would have thought about genomic mapping for preterm labor? And our next speaker is equally, he's got an impressive profile. And to introduce this, I need someone as impressive as him, Dr. Kamini Rao, to do the honors. I'd like my mentor, Dr. Kamini Rao, who is the organizing secretary for the conference, to introduce Dr. James Padbury.
Hi, James.
Good evening.
Good evening. We have Dr. James Padbury, who's the O. Zophi Endowed Chair at Brown University and Pediatrician in Chief at the Women's and Infants Hospital. He's the director of the Rhode Island Center for Clinical and Translational Research, and his medical and postgraduate training were received in the University of California. We also know that he has also been given grants for this particular thing, the March of Dimes grant, which is a very prestigious grant given for translational research into genome mapping of those with preterm labor. Who would have ever heard that preterm labor would actually reach to this stage? But we have Dr. James Padbury to speak to us on this. Thank you very much. Welcome, Dr. James Padbury.
Thanks so much. I hope you can hear me well. I can see and hear you all very effectively. So what I'm going to do today is, this is an outline of the things I'll present, a brief primer on genetics of preterm birth and the genetic architecture of complex diseases. I'll review our approach to DNA sequencing to address this problem. I'm going to describe some fairly sophisticated computational approaches to the analysis of the genetics of complex diseases, and then discuss the generalizability of these approaches to other complex diseases. So there are numerous risk factors associated with prematurity. We've already heard many of these this morning: a prior history of preterm birth, stress, infection, inflammation. Racial groups have higher risks, trauma, adverse sociodemographic factors. The leading etiology is idiopathic, but genetic factors do play an important role. Classical estimates from twin studies suggest that the heritability of preterm birth is around 40%. That's also supported by family studies where the risk within families is greater. So genetic and epidemiologic studies, very interestingly, put all the risk on the maternal side. So a woman who has herself born premature, if she has preterm infants, the female offspring have a greater risk of having additional preterm infants, but not her male offspring. How did we identify preterm birth associated genes? So a number of years ago, we developed a database for the genetics of preterm birth. As you can see, we used natural language processing, semantic data mining to go through the entire world's literature of published data on preterm birth. We looked through public expression databases, linkage data, and we generated a nominal set of genes that were associated with preterm birth. Turns out of the 20 million records in PubMed, 30,000 have are linked to preterm birth, but 1,200 of those have genetic associations associated with preterm birth. We had to read all of them in order to curate this database and come up with this set of useful genes for our future studies. We next used these gene sets to examine a genome-wide association study on preterm birth. There were, in our study, there were 2,000 preterm and 2,000 control patients. This was a collaboration with the Danish National Birth Cohort and the Gene Environment Association studies at NIH called Geneva. These patients were all genotyped on an Illumina genotyping platform with around 600 to 900,000 SNPs on each chip. Interestingly, no significant associations were found in this genome-wide association study. That doesn't mean this disorder is not genetic. That means this disorder is not associated with variants in common variants and single gene variants. This is a complex genetic disorder. It's not, if it was Mendelian, we would know this. it's a complex genetic disease. So the genetic architecture is that genes and variants in genes and pathways that are linked biologically are what are sufficient to give rise to the disorder. So a number of variants in a given pathway can give rise to this disease. So we reanalyzed the genome-wide association study. Instead of using univariate associations, we did what's called gene set enrichment. So you can see the networks on here. We looked for variants occurring simultaneously in given networks, given biological pathways that we know exist, and we looked for their differential abundance between cases and controls. This has become a very contemporary approach to complex disease genetics. So remember that, gene enrichment association. We used our gene sets from the database for preterm birth, and you can see this is a Manhattan plot, and it shows that there are a number of significant networks. You can read those along the horizontal axis. On the vertical axis is the negative log P. So the more statistically significant an association is, the higher the column. The genome-wide cutoff is the yellow bar. So you can see there were a number of gene sets associated with preterm birth exclusively with the gene set from the database for preterm birth. There were gene sets defined by both the database of preterm birth, and when we did a genome-wide association, the database for preterm birth genes are shown in dark blue. The genome-wide associations are shown in open columns, and you can see the value of the database we built. The genes used the genes from the database for preterm birth had many more significant associations than the genome-wide alone. However, there were also some gene sets identified only by the whole genome analysis. So important observations and the value of developing specific gene sets for individual disorders like preterm birth. This shows both this analysis. There were a total of 329 genes identified in these in this gene set enrichment analysis of the GWAS. This data were published in Genomics. On the cover of this, you can see the aggregate networks and pathways associated with preterm birth. I don't expect you to be able to read those networks and pathways, but I want to call out an interpretation for people who are, for whom this heavy-duty genomic stuff is a little new to their thinking. This is the network and pathway of genes and gene sets for preterm birth. Imagine these are stones in a river and you want to cross from the left side of the page to the right side of the page. You can get across the river jumping on any group of stones. Any one of these pathways is sufficient to give you the disease phenotype. I've circled a few of those just to provide an example. We targeted this group of 329 genes for deep sequencing. The way we carried out the sequencing was captured technology, fragment the DNA, pull down the genes in the regions surrounding the variants that we were interested in, and then subject them to deep sequencing. Our strategy was further enhanced by saying we should only sequence extreme phenotypes for preterm birth. So what is an extreme phenotype for preterm birth? You heard a classification discussed earlier. We reasoned that since the incidence of prematurity has been rising over the last several decades, but the biggest increase has been in late preterms, that's an environmental cause, that's not a genetic cause. The incidence of preterm birth in the very early gestations has been steady over the last number of decades. So we chose as our extreme phenotype only infants under 34 weeks, without drug abuse, without twins, without preeclampsia. And we focused heavily on those with a family history of preterm birth. So you can see of the almost 200 patients that we've sequenced, we had 12 patients with 3 generations of preterm birth, 34 patients with 2 generations of preterm birth, And you can read these data. Focusing on a prior history of preterm birth leverages the likelihood of a genetic cause. I want to say that these were very carefully phenotyped. Genetic pedigrees were taken on all of these patients, and importantly, the same genetic pedigree and careful ascertainment was used to identify the controls as not having delivered preterm and having no family history of preterm. As you can see, we had 2-to-1 cases versus controls. Of course, the gestational ages were different between the cases and controls. This shows the racial and ethnic background on the cases and controls. That we matched patients for race, ethnicity. You can see we did a good job matching, but nonetheless, there was still significant population stratification that I'll come back to. This is our sequencing approach. I already mentioned the custom bead capture. We used the Illumina sequencing platform. This was exceptionally good sequencing, 20 million reads per patient with extremely good sequence quality data. This is the workflow for the sequencing pipeline: DNA fragmentation, library preparation, sequencing and base calling, and then bioinformatic analysis. This is the analytical approach. We first identified variants and then called them whether or not they were pathogenic. Genotype testing refers to comparing the incidence between cases and controls. We were interested in variants that were not only likely to be predicted to be pathogenic, but that were differentially abundant between cases and controls. If we found common variants in both cases and controls, we filtered those out of our variant pipeline. And you can see the remark correcting for population stratification. I'll show you that on the next slide. And then we carried these through another gene set enrichment and multivariate analysis. These are the primary sequencing results. Almost 40,000 variants identified in these 200 patients, and you can see their distribution across genomic regions. Because we were capturing some intronic regions, you can see that there was an abundance of variants in those regions. This is principal component analysis. The colored circles refer to the different race ethnicity groups, and you can see there was significant clustering around race ethnicity. If we didn't take this into account, we could be attributing a variant to preterm birth when really the variant was just as a marker for their race ethnicity group. So the way you control for that is using something called Eigen Strat testing. And I love statistics and numbers. I think the audience can tell. So you can see in the column over to the far right, the final F statistic for significance when after correction. So I've highlighted the ones in red where there was significant improvement in the statistical validity of the variant after correcting for population stratification. So it's extremely important in genetic diseases to take this into account, even after we matched during our enrollment. All right, gene set enrichment to find what clusters, what networks of genes that we found between these cases and controls. We built a new software. This is available on, as you can see, on bioRxiv for anybody who wants to follow through with this. We built a network for each patient. Then we compared the networks, similarity matrix of those networks. And then we built a Dendogram and visualizing networks for the outcome of the statistical comparisons. I'm going to show you several dendrograms, which are a way to demonstrate similarity among patients. It's used commonly for taxonomic classifications of different genuses and species. So I know you can't see this. This is the full dendrogram of our results. But if I blow it up, you can see that there's significant clustering between these patients. Each one of the horizontal lines represents an individual patient. The cases are shown in yellow, the controls are shown in blue, and you can see that there's significant clustering of these patients. Remember, they're clustered because of the similarity of their networks, and the disease prematurity is due to a network, not a single variant. This is seen better if I convert the dendrogram into a circular dendrogram, and I show you which of the clusters which were indeed significant. So you can see in the top right corner that there are cluster 86 was dominated by cases. There was a greater number of cases than controls in cluster 86. Similarly, there was a greater number of cases than controls in cluster 126. I've outlined these in dark dotted lines just so you can see how they relate to one another on this dendrogram represented as a circle. And of the 190 patients, we were able to assign 66 of these patients in total to a significant cluster. These are outputs, these are the networks that underlie those clusters. So in panel A, the case-dominated cluster number 120, you can see the genes that are associated and shared by those patients. The ones that are unique to this cluster are shown in blue. Whenever I give this talk, people raise their hand and they ask me about, what about IL-4? What about individual genes? They have their favorite candidates. And I always point out to them, this disorder's genetic architecture isn't due to a single gene, but it's due to variants in pathways and clusters of genes. But nonetheless, I think looking at these individual genes is important. The other case-dominated cluster is shown in panel B. Perhaps a better way to think about this disorder is to subject these significant clusters to pathway analysis. And I've shown you the top diseases and pathways associated with cluster 120. So you can see inflammation, inflammatory disorders are among the pathways that are highly represented, cancer, reproductive system diseases. This was done with ingenuity pathway analysis. And you can see the kinds of molecular and cellular functions that are underlying this. Cell morphology, cell-cell signaling is extremely prominent in cell development. This is for the case-dominated cluster 120. Similar results are seen for cluster 86, though there are unique genes to each one of these. Again, inflammation is a highly represented disease process, as is tissue injury, cancer, and of the cellular functions, again, cell movement, cellular development, signaling is very prominent. So what I've shown you is that prior genetic analysis and filtering of both the patients and gene targets improves the likelihood of finding otherwise difficult to find rare variants. So our prior genetic analysis was building data sets in order to know where to look for disorders, and then the prior filtering of the patients is leveraging extreme phenotypes, both in terms of their stage of gestation and the family history of preterm birth. And we would conclude that using large data sets and layered approaches like this with pathway analysis, gene set enrichment, and metagenomics, we've identified genes and networks and pathways associated with this complex disease. But this approach is generalizable to the analysis of other complex diseases. If you look at our work, you can see that we've developed a database for preeclampsia as well, and I actually have very similar, exciting data on the genetic architecture of preeclampsia. Not enough time to share with you. And no one works alone, including me. These are members of my lab, people from other universities with whom we've collaborated, and of course, funding. And thank you so much for your attention.
Thank you very much, sir. Thank you indeed. And we all know it's almost around 1 a.m. there, and we appreciate your efforts to keep yourself up and keep us engrossed in this talk that you gave. We are open for some discussion and questions from the audience. I think we still have 10 more minutes. Any questions from the audience? I think we have someone there. Can we get a mic there?
Thank you, Doctor, for that wonderful talk, going into the details of the preterm.
Virtually, Dr. Padbury.
Check. Can you hear us?
I hear you now.
Yes. The question is, after all this molecular research, now, how is this genomic mapping going to help the physician or the clinician to, risk modulate a patient with previous history of preterm birth or who is at risk for preterm birth?
So there won't be a single genetic test to predict preterm birth or to a single gene test to predict preterm birth or to identify someone who's comes in and we're uncertain whether they're in preterm birth. But you saw that there was a modest number of genes in the network and pathways associated with preterm birth. So a multiplex test incorporating 10 or 15 of these genes that we can associate in this way may actually allow us to have a rapid PCR-based test for whether or not someone who presents with uncertain whether or not they're in preterm labor, whether they have genetic risk factors, or whether there's prediction before pregnancy. The other very contemporary genomic approach to disease management is drug repurposing. So we can look at expression data for these genes and identify drugs that can modify the expression profiles in a way that will attenuate the consequences of these pathways. And that's something we're working on already with preeclampsia. So I know this is unfamiliar material to most people. This work was just presented at the American Society of Human Genetics. So it is complex, but it's very contemporary and robust way to get at something that's been enigmatic up until now.
Any more questions?
Dr. Padbury, before we actually, there are plenty of questions, but I know because of paucity of time. So what you're trying to tell us is you're going to map genes which are probably attributable to a particular pathway which may lead to preterm labor. For instance, there can be a genetic predisposition to inflammation, and inflammation can lead to preterm birth, and you will modify inflammation when you identify these women who would be predisposed to this particular trigger?
So I very much want to hear you, but I could not hear that well.
Is it that you are identifying genes which could probably trigger inflammation or a particular pathway which could predispose to preterm birth?
Yes, inflammation was a prominent disease process, prominent descriptor for the pathways in both of the case-dominated clusters. However, you and the other speakers all know that strategies directed solely at inflammation have not been successful in preventing preterm labor or in preventing preterm birth. That includes antibiotics, anti-inflammatories. So this is a disease process that includes more than just inflammation. But I do think that inflammation is, we found it in our gene sets as well. So it's an element, but it's neither necessary nor sufficient to give rise to preterm birth. There are other pathways involved.
So one last question. Since we are talking about the genetic genomic mapping, this will vary according to ethnicity as far as I have a limited knowledge of genetics, but then yes, it is going to vary according to ethnicity. So in Indian population or Southeast Asian population, we would need a different set of genomic mapping in them, or the same thing what is done would apply to them. I would like to know, sir.
So as we acquire more cases, we're going to be able to say whether or not the pathways vary specifically by race ethnicity. We haven't done that with this data set yet. As I said, we had about 130 preterm birth cases that had strong family history of prior preterm birth, and 60 controls that had no family history. What you all could help me with is identifying another set of patients and samples carefully phenotyped in this way, get us a 0.5 ml of blood, and we can build an Indian cohort.
That'll be really exciting, sir, to liaise with you, and
collaboration.
That'll be really great if we could do that. I think Dr. Revati and I are already excited about it.
Thank you so much.
It's particularly important if we can focus in on cohorts of patients where there's genetic leverage. So if indeed there are population groups in your country that have a greater risk. We certainly attempted to enroll as many African Americans as possible because their risk for preterm birth is biologically determined. It's not due to sociodemographic factors. So we are always looking for important cohorts like that. And clinical phenotyping is as important as this fancy sequencing and mathematics.
Exactly, sir. Exactly. It's not a replacement to the clinical judgment and phenotyping. Thank you very much, sir. We appreciate the efforts taken by you to spend your valuable time with us at this late hour. Thank you very much from all of us here in India.
And you could participate
pleasure.
at any point of time, sir. Considering that you're logged into the discussion till the end, you could actually contribute your views at any point of time during the discussion time.
I think that was indeed an exciting session for all of us. We move on to our next speaker, who's equally dynamic, and the topic cannot be more enigmatic than predicting preterm birth in multiple pregnancies, and it's a multivariate model. Dr. Gayatri Karthik, we welcome you, ma'am. She's a senior consultant obstetrician in Manipal Hospital, and her key interest areas are robotics, infertility, and high-risk pregnancy, of course. We welcome you, ma'am.
If I move these slides.